Okay, so here we are, multivariate community analyses. We're going to be doing some things this week where we're looking at the entire community in a location. In this case, maybe we're looking at all kinds of stream insects in a plot. You might be looking at all kinds of plants in a plot, all kinds of trees in a stand. You get the picture. So we're looking at lots of different things. So just to kind of put this in the context of where we've come so far, most of the quarter we've spent looking at single variable univariate statistics with continuous or categorical Y variables and continuous or categorical X variables. So hopefully you're feeling pretty comfortable with these this kind of four panel plot. Now we've also expanded this. We've moved into multiple X's where we've dealt with two-way, three-way ANOVAs. We haven't really gone into multiple linear regression, but you could imagine multiple X's influencing variation in Y. That would be multiple linear regression. We've talked a little bit about multi-way contingency tables and CART modeling, so classification and regression tree modeling. And now we're going to move into multiple Ys. We're going to talk a little bit about what's called MANOVA, multivariate analysis of variance, permutative MANOVA, ANOSIM, which is an analysis of similarity, MRPP, we'll get there, and ordination. And then there's another thing that you could add, which is called structural equation modeling. There's a lot you could add, kind of on the outside of this four-panel box, but just kind of to place this into the context of where we're headed. We're headed off into this direction with multiple Y variables. Okay, so multivariate data sets, that's what this means, having multiple Ys. And we've already kind of talked about a few of them, but they really can be rich and diverse, and it can cross disciplines beyond just ecology. So, for example, we can think about diverse communities of people, plants, insects, microbes, birds. We could think about assemblages of chemicals. What are all of the phytochemicals in a plant? We could think of assemblages of variables, genetic markers, assemblages of taxonomic attributes or traits that make up good species. We could think about assemblages of survey responses or values or opinions. And you could think about assemblages of answers on tests. So you can actually ordinate classroom data as well. So really any data set where you have many, many Y variables. And that's where we're headed. All right. So we're not talking about data sets with many X's. We're just going to talk about, well, I guess I take that back. You can have categorical X variables, and you can also have continuous X variables that explain some shift in the community structure. So we're going to kind of focus mainly on the fact that we have many Ys, but you can also have many X's. So when you organize your data, or if say you're collecting data in the field, you want to keep your samples separate. Do not pool data from your samples. So you'd want to enter your data like this. So you have sample number like Snyder 1 versus Snyder 2, and then you'd have which species did you find in the plot and how many of them. So you have these columns, the sample ID, the species that you encounter, and the abundance of each species. And if you enter your data in a list structure like this, you can easily convert it into a matrix. And I didn't know that when I was doing my PhD. I would have probably saved myself about 7 months of my life if I had known about pivot tables. So once you have this long list of species and abundances, then you can use what's called a pivot table in Excel to transform your list into a matrix. And now you can see, instead of a list, I have my two samples as rows, and then I have all of my species as columns in my matrix. And this is how you want to organize a community matrix. You're not going to have to do this for the lab, but I just wanted you to know a few little details about data management. So you might end up with a community matrix like this. Again, you have some code for the plot or the sample as a row, and then you have some code for all of your species names across the top. And then you have all of the data in here. And you can see there's a lot of zeros, which is fine. Every cell needs to have some value. You can't have any blanks. All right, so some basic community analyses before we get started. We've talked about some of these already in this program. Species richness. Once you have your data in a community data set in, you can calculate things like species richness. You can calculate species abundance. So you can look at a single species at a time. You can look at species evenness, and then you can combine richness and evenness and measure some kind of diversity index. So these are all really great things that you can do as a kind of a simple first step for analyzing some community structure. Then you might be interested in a treatment effect on your community. So what can affect the structure of a community? Well, all kinds of things. And you could look at each of these effects on like a single species at a time. You could use ANOVA or a t-test or a two-way ANOVA or a regression to look at how does the number of mosquito larvae change across some kind of a gradient, or are there more mosquito larvae in natural ponds versus mitigated wetlands, right? So you could start imagining how you can look at just single species using the tools that you already have. You could also use these same tools that you've already learned, t-tests, ANOVAs, regressions, to look at the effects on species richness, total abundance in your community, or diversity indices in your community. Okay, so now we're still talking about all these things that you already know how to do. I just want you to kind of get a sense of how much you've learned so far. All right. The next step, though, is to really examine the whole community, how the whole community is structured. So whole community analyses involve what are called ordination methods. And there's a few different things we're going to learn about. One is called a multi-response permutation procedure. Another is called an analysis of similarity, or ANOSIM. And the last is called a permutative multivariate ANOVA, or a PERM-ANOVA. And then there's some other really cool things that you could add on if you wanted to do more advanced work, things like cluster analysis, factor analysis, Mantell tests, indicator species analysis, a few other things. So I'm going to stop here and give you a little break, and then we'll move on to ordination and its possible uses.